Okay, so let's look at the C4 pathway. So the C4 pathway of tropical plants allow for more efficient photosynthesis by concentrating CO2. So unlike the Calvin cycle, because oxygenase activity of rubisco increases more rapidly than the carboxylase activity at higher temperatures, some plants concentrate CO2 in photosynthetic cells to reduce photorespiration. So plants that perform C4 synthesis can keep their stomata closed a lot more than C3, or plants that use C3, because they are more efficient in incorporation of CO2 and minimizing water loss. So, for example, right, corn plants or even cacti are usually in environments of high heat. So they would use C4 to keep their stomata closed to minimize the loss of water, whereas plants in the tropics, such as like trees in the rainforest, they would use C3 because they don't need to worry about keeping their stomata closed because they're constantly getting that. They're constantly replenishing that water. So C4 pathway is really important for saving up water for photorespiration. So the C4, also known as the Hatch-Slack pathway, is called this because it involves 4-carbon intermediate. Okay? So let's draw the pathway and talk a little bit about how this pathway works. So you have 2 types of cells. So I'm just going to draw them like this. So this is the air. This is the mesophyll cell. And these are the bundle sheath cells. Okay. So let me highlight these words. Okay. So the mesophyll cell is where photosynthesis occurs. And this specific cell needs CO2, and it can receive that CO2 from the air. Versus the bundle sheath cells, they're cells that surround pipes of plants that distribute fluid. These cells cannot have access to the air. So they're found a lot deeper. Okay? So we have CO2 coming in from the air. And this CO2 can only be fixed by PEP. And we'll talk about the differences between the CO2 and the CO2 that's found in the bundle sheath cells. So CO2 comes in. Let me just draw this region right here. Okay, so CO2 comes in. And you also have phosphoenol pyruvate, also known as PEP, which is a 3-carbon. So you have CO2, which is a 1-carbon, and you have PEP, which is a 3-carbon. And the two come together, and that's how they form oxaloacetate, which is a 4-carbon. And oxaloacetate converts to malate. Okay? And malate can actually be transported to the bundle of sheet cells, where it can release a CO2 and make pyruvate. And this CO2 is really important for entering the Calvin cycle. Okay, so we see a CO2 here and a CO2 here. And the difference between these two CO2s is that the first one that's coming in to the mesophyll cells can only be fixed by PEP. Whereas the one that's in the bundle of sheets can now fix the CO2 with ribulose bisphosphate with Rubisco, producing sugars. So this CO2 is the one that you see in the Calvin cycle, whereas the one here cannot be fixed by Rubisco. And so the first CO2 is facilitated by PEP through the carboxylase enzyme. Okay, and it's can only fix carbon. Specific enzyme can only fix carbon, not oxygen. So here you have the facilitation of PEP instead of Rubisco. Okay. So that's what you need to know for the C4 pathway. Also, knowing the difference between C4 and C3 is important. So C3 plants are found in most plant species because there's usually access to water, whereas C4 are in more agricultural plants because they need to maintain their water loss. And the water loss is due to evaporation, which is problematic for a lot of plants that live in the desert, which is why they need to keep their stomata closed. Okay. Also, Dr. Thomas talks about CAM. I will put it in this corner here. And CAM is important for trapping CO2. So plants using CAM allows the stomata in leaves to remain shut during the day. And this allows the reduction of evapotranspiration. And the stomata opens throughout the day, and CO2 required for Calvin cycle is concentrated using the C4 pathway. So CAM is actually what allows for the stomata to shut, and the C4 pathway is what allows the CO2 that is needed to be incorporated into the Calvin cycle.